So in this talk, I'm going to briefly summarize some results from a side project that I became involved in earlier this year. And this was focused on angulate herpes virus 1, which is a virus of freshwater eel species. In our lab, this is primarily studied by Hayin and Natasha under the guidance of Professor van der Plaschen. And they've generated a substantial amount of full-length genome data, giving us a great opportunity to make the first comparisons between angulate herpes virus 1 and that of its closest relatives in the Cyprinovirus genus. Within this genus, angulate herpes virus 1 is rather unique, of course, in the sense that it infects angulate hosts as opposed to all other members which infect cyprinid hosts. These two groups of hosts are very different in terms of physiology and behavior, of course, so we wondered if there were very different forces at play in the evolution of their respective viruses. So first of all, we looked at relative species clade divergence times. Using 4 core genes common to all of these viruses, we used a Bayesian approach to make many phylogenetic estimates and used a subsample of these to construct a maximum clade credibility tree with branch lengths representing time. And this indicated that angulate herpes virus 1 diverged earlier than the other species clades. We then looked at differences in evolutionary rates. In this tree here, based on the same 4 core genes, with branch lengths representing substitutions per site this time, you can see, of course, that angulate herpes virus 1 has accumulated less substitutions than its relatives since they diverged from their most recent common ancestor. But we wondered, are these differences in any way significant? So we used Tajima's relative rates test to systematically identify instances of violation of the molecular clock hypothesis between different parts of the phylogeny. And we did, of course, find such violations between different parts, but in particular, where angulate herpes virus 1 lineages were used in our pairwise tests, indicating that it may indeed have evolved under a much different, evidently slower rate based on comparisons between these shared core genes. So if these viruses have cyprinid hosts diverged more recently, after evolving much faster into the species that we know today, we wondered if we would see corresponding differences in selective pressure at equivalent periods in their evolution right before species clade divergence. So we investigated this using branch site models, and this revealed strong evidence of positive selection taking place in the DNA polymerase and helicase genes within the Cyprinivirus genus, including cases of convergent evolution in CYHV1 and CYHV2 DNA polymerase genes. Notably, one of these sites under selection in CYHV2 displays conservation of structural location across the genus, which is quite interesting. But the important point here is that these significant positive selection signals were only detected in faster evolving viruses of cyprinid fish, with nothing significant detecting in angulate herpes virus 1 sequences. So we see a trend emerging here indicating that angulate herpes virus 1 is somewhat different. It appears to have diverged earlier under a lower evolutionary rate and displays at best lower signals of positive selection. And this may be linked to the expected differences in epidemiology, because don't forget, we're talking about a very different host species here, with very different behavior and very different life cycle. So to sum things up here quickly, eels are much more solitary creatures, and their reproductive behavior is less conducive to sustaining a continuous viral transmission chain, given that they die after spawning. Cyprinid fish, by comparison, are quite the opposite in both of these aspects. Overall, eels engage in much more long-term social distancing, and this may, of course, result in less opportunities for transmission, and may indeed have an impact on the evolutionary kinetics of angulate herpes virus 1. But so far in our comparisons of divergence times and evolutionary rates, we've been describing this indirectly, in relative terms, using arbitrary units. So we wondered if we could derive the same estimates in absolute units. So we explored the possibility of deriving such estimates by calibrating timelines in trees with events that can be dated, and using this to extrapolate the ages of other nodes of interest. This is known as node calibration, and by extension, of course, we can estimate evolutionary rates. Given that herpes viruses undergo co-speciation with hosts, we explore the use of host divergence states to calibrate nodes in our tree. However, the use of hypothetical co-speciation assumptions is highly speculative in this case for several reasons, so we need to further refine these models as more data emerges. Alternatively, going forward, to avoid such speculative assumptions, we're going to explore the calibration of our trees using known sampling dates of sequences, and then extrapolate back to derive short-term estimates for divergence time, and of course, by extension, short-term estimates of evolutionary rates. In terms of rate, what's being measured here is more representative of non-lethal mutation rates, which is different to substitution rates being measured with node calibration, which are themselves more representative of the rate at which these mutations become fixed in populations over time. But I'd argue that this information on non-lethal mutation rates may be of much more practical use in this case. Also, interestingly, short-term mutation rates may also help us understand why we see much lower genetic diversity among these fish herpes viruses compared to what we see in mammalian herpes viruses. So to conclude, Angular herpes virus 1 appears to have evolved differently than its closest relatives when we compare them in terms of these four core genes. Deriving absolute estimates of divergence times and evolutionary rates using node calibration is a bit difficult at the moment, considering the uncertainty surrounding co speciation events and dating. But for many reasons, collaborative efforts towards generating short term estimates would be very useful for the field. So thank you very much for your attention, and thank you to all the team in the lab.